In the shadowy landscape of neuro-oncology, where the diagnosis of glioblastoma multiforme, or GBM, has long been synonymous with a countdown, a revolutionary beacon has been lit. It does not come in the form of a scalpel, nor a radiation beam, nor a pill. It comes as a fluid, a shimmering, golden-hued injectable solution that, once inside the human brain, performs a feat of molecular alchemy. It transforms from a liquid into a soft, living circuit, a bioelectrode that seeks out the enemy with the precision of a guided missile and the conductivity of a metal wire. This is the dawn of injectable bioelectrodes, a technology that marries the cutting edge of nanotechnology with the primal power of bioelectricity. To understand the magnitude of this solution, we must first deeply respect the problem, because glioblastoma multiforme is often called the terminator of cancers for a reason. It is not a solid lump that can be simply cut out. It is a web. Its tentacles, known as phyla, infiltrate the healthy brain matter, wrapping around blood vessels and neurons like a parasitic vine. A surgeon can remove 99% of the visible tumor, but the microscopic 1% remains, hiding behind the blood-brain barrier, waiting to regrow. This barrier is the first wall of resistance. It is a physiological checkpoint that denies entry to 98% of small molecule drugs and 100% of large molecule therapeutics. It protects the brain from toxins, but it also protects the tumor from chemotherapy. Then there is the second wall, the immune-privileged environment. The brain is an immunologically cold zone. It has few resident T cells, and tumors like GBM actively suppress the immune system, cloaking themselves in a biological invisibility shield. Finally, we face the wall of physical inaccessibility. Deep-seated tumors are often inoperable. Traditional electrodes used for deep brain stimulation or electrocautery are stiff, metallic needles. Shoving a rigid metal rod into the soft, tofu-like consistency of the brain causes inflammation, scarring known as gliosis, and damage to healthy neural circuits. The current standard of care, which involves surgical resection followed by radiation and temozolomide, has stalled, with median survival hovering stubbornly around 15 months. The medical world has been waiting for a weapon that can bypass the blood-brain barrier, spare healthy tissue, and kill the resistance. That weapon is the injectable bioelectrode. The breakthrough, spearheaded by researchers at institutions like Lund University, centers on a concept called in situ self-assembly. The goal was to create an electrode that isn't built in a factory, but builds itself inside the tumor. The magic lies in a two-component system, referred to in recent literature as the A5 ETE PC complex. The first component, A5, is a peptide-based biological scaffold. Think of it as the skeleton. It is designed to be fluid at room temperature or when mixed in solution, but upon contact with the specific ionic environment and temperature of brain tissue, it begins to self-assemble into a fibrous hydrogel. The second component is ETE PC, the conductor. This is a conjugated polymer variant. Alone, it is just a chemical, but when mixed with A5, it utilizes the peptide scaffold as a template. When a neurosurgeon injects this solution into the tumor cavity or directly into a non-resectable tumor via a thin needle, a fascinating chain reaction occurs. First, the fluid flows effortlessly into the irregular shapes of the tumor cavity, filling every crevice and microscopic fissure where cancer cells hide. This creates a conformal contact that is impossible with rigid metal electrodes. Then, within minutes, the A5 component reacts to the body's physiological conditions. It locks into a hydrogel, a soft, jelly-like substance that matches the Young's modulus or stiffness of the brain tissue. This eliminates the mechanical mismatch that causes scarring. Simultaneously, the ETE-PC component begins to crystallize and polymerize along the A5 fibers. It forms long conductive dendrites or pathways throughout the gel. The result is a fully functional, highly conductive electrode that has grown into the exact shape of the tumor. It is soft, biodegradable, and intimately connected to the cancerous tissue. Once this bioelectrode is formed, the treatment begins, and the method of choice is irreversible electroporation, or IRE. Unlike thermal ablation, which burns the tumor, or cryoablation, which freezes it, IRE is non-thermal. It relies on the physics of the pore. Every cell is wrapped in a membrane, a lipid bilayer that acts as a skin. This membrane maintains the voltage difference between the inside and outside of the cell. When high-voltage, ultra-short electric pulses are delivered through the injectable bioelectrode, the external electric field creates a voltage potential across the cancer cell membrane. If the voltage exceeds a critical threshold, the lipid bilayer is forced to rearrange. Nanoscopic pores or holes blast open in the membrane. In reversible electroporation, which is used for gene delivery, these pores close back up. But in irreversible electroporation, the pulses are strong enough that the pores act like permanent wounds. The cell leaks its contents, its internal balance collapses, and it dies. The beauty of IRE lies in its selectivity regarding the structure of tissue. It offers collagen-sparing benefits. The protein matrix that makes up blood vessels and nerve sheaths is not made of cells, it is a structural lattice. IRE does not affect collagen. This means a tumor wrapping around a major artery can be destroyed without popping the artery. Furthermore, while neurons can be affected, the soft bioelectrode allows for such precise field focusing that the kill zone can be sculpted to the tumor shape, sparing adjacent healthy brain matter. But if IRE were simply killing cells, it would be just another form of surgery. The true power of the injectable bioelectrode lies in how the cells die. This is what we call the immunotherapy bonus. Chemotherapy often causes apoptosis or silent cell death, where the cell quietly implodes and is cleaned up by the body without a fuss. IRE, however, triggers immunogenic cell death, or ICD. This is a loud, messy death that screams for attention. When the cancer cell membrane is perforated by the bioelectrode, a cascade of signals is released. First, the cell dumps its energy reserves, ATP, into the extracellular space. To the immune system, free-floating ATP is a find-me signal. Second, a protein usually hidden inside the cell called calreticulin is forced to the surface. This acts as an eat-me signal to macrophages and dendritic cells. Third, the nucleus releases high-mobility group box 1 protein, a potent danger signal that activates the innate immune system. Glioblastomas are notorious for being cold tumors, meaning they are invisible to T cells. The IRE treatment acts as a vaccination in situ. Dendritic cells swarm the treated area, attracted by the ATP. They consume the debris of the dead tumor cells, including the unique cancer markers or neoantigens. These dendritic cells then travel to the lymph nodes and present these antigens to T cells, effectively identifying the enemy. The newly trained T cells, specifically CD8 positive cytotoxic T lymphocytes, rush back to the brain. They cross the blood-brain barrier and hunt down not just the main tumor, but the microscopic gorilla cells that drifted away from the primary site. Recent studies in animal models have shown that this abscopal effect can clear secondary tumors and prevent recurrence, something surgery alone can never do. While the conductive hydrogel A5 ETE PC is the frontrunner, it is part of a broader ecosystem of nanoparticle therapies emerging right now. We are looking at Magnetoelectric Nanoelectrodes, or MENs, developed to solve the wiring problem. MENs are nanoparticles that convert magnetic fields into electric fields. Instead of injecting a gel connected to a wire, you inject millions of these dust-sized particles. The patient wears a magnetic helmet, similar to an MRI coil but weaker. The magnetic field travels harmlessly through the brain, but when it hits the nanoparticles inside the tumor, the particles vibrate and generate a local electric charge. This allows for deep brain stimulation without any implanted wires or catheters. A more exotic approach involves bio-nanoantennae, which are gold nanoparticles coated with redox active molecules. These particles are tuned to interact with the electron tunneling mechanisms of the cancer cell's mitochondria. When stimulated by an external electrical field, these antennae hijack the cell's own electrical signaling, commanding it to commit suicide. This is being hailed as the first quantum therapeutic. So what does this mean for a patient, let's call him John, diagnosed with a treatment-resistant brain tumor in the near future? Imagine John has a recurring glioblastoma near his speech center. Surgery is too risky. Under local anesthesia, a thin needle is guided by MRI to the tumor center. The surgeon injects the A5 ETE PC solution. It flows into the tumor, filling the irregular cavity. A thin wire connected to the gel is activated. John feels nothing. The IRE pulses are delivered in microseconds, too fast for nerve pain receptors to register. The electrode is left in place. Over the next few weeks, it can be reactivated for booster sessions if needed. After 3 months, the electrode, being biodegradable, dissolves harmlessly. By then, the tumor has been necrotized, and John's own immune system is patrolling the area. This is vastly different from current electrotherapies like Optune, which requires patients to shave their heads and wear a cap of ceramic discs 18 hours a day for months to create a low-intensity field that merely slows cell division. Injectable bio electrodes are high-intensity periodic treatments that deliver killing power directly to the source. However, challenges remain before this becomes the gold standard. Delivery precision is crucial. Ensuring the hydrogel stays exactly within the tumor and doesn't leak into ventricles is a priority, leading to tests on new viscosity-tuned polymers. There is also the risk of an immune storm. While activating the immune system is beneficial, too much activation in the brain can cause edema or swelling, so steroid management protocols must be refined. Regulatory pathways are also complex, as this is a combination product involving a drug, a device, and a biologic, though the orphan drug designation for glioblastoma may fast-track these trials. In conclusion, we are witnessing the end of the cut and poison era of oncology. Injectable bioelectrodes represent a shift towards smart therapies that respect the biology of the patient, while ruthlessly targeting the pathology of the tumor. By transforming the tumor itself into the electrode, we deny the cancer its greatest advantage, its location. For the thousands of patients facing the dark diagnosis of treatment-resistant brain tumors, this golden fluid offers something that has been in short supply. Not just a treatment, but a cure driven by the body's own awakened defenses. The future of brain surgery is soft, injectable, and electric.